Okay, so ladies and gentlemen, you are all welcome, and I hope we are all ready to start the class. And I think if you could remember, I sent you about so far about 4 videos link, and that is where we are going to continue with. So today's class will be based on questions and answers. So I send you about 3 video link to watch. So therefore, the major discussion will be based on question and answer. So therefore, but I hope you can all hear me. Are you with me? Can you hear me clearly? Yes, good evening all. Can you all hear me? Okay, excellent. So as I was saying, we are going to discuss more on those videos that I send you. So I will be asking you questions on that. So now I want to start by asking you, what is biochemistry? What is biochemistry? So I'm not sharing any slides today. As I said, we are going to revise and discuss on those videos that I send you the link. So therefore, if you have any questions, you can ask me. So therefore, I will be asking you and you will be asking me. But let me answer one of the questions that I sent today early in the morning. That I asked a question that why, in terms of the birth rates, the number of men are less than the number of women generally. That is usually happen. If you now look at how the population of the world is going, you will realize that when there is actually whenever there is giving birth of babies, you will realize that about 70 to 80% of the babies now that we are giving birth are girls. So what is the question? Sorry, what is the reason behind that? So seriously speaking, ladies and gentlemen, I think if you could remember, I told you that we have uh, X chromosome, XX chromosome for women, while XY chromosome for men. So generally speaking, human species, we are in danger. Remember, from your basic knowledge of biology, we have what we call evolution. Evolution is a process whereby a living organism change with time. So generally, organisms or living organisms are changing with time. So that is what you call evolution. So currently, humans are also going through that evolution. As of now, that Y chromosome that you are seeing is now changing through evolution to X. So the Y chromosome is changing to X. So a time will come, if care is not taken, because the way the evolution is taking human species, there is a likelihood that will stop giving birth to boys, because the Y chromosome is now changing to X. Remember, we have 2 types of sperm. Who can tell me we have 2 types of sperm? We have 2 types of sperm. So who can tell me what are the 2 types of sperm that we have? And I think I discussed this in one of my videos. We have 2 types of sperm. So who can tell me what are those types of sperm? What are the types of sperm? Yes, what are the types of sperm? Yes, Destiny, I'm serious. That is why now if you check, in most cases, you will see that the number of the babies that we usually give birth, most of them you will realize that they are girls. So this is actually due to the problem of evolution. Excellent. It's not X chromosome sperm. We call it X sperm and Y sperm. So whenever there is, no, it's not mortal and non-mortal. We have X sperm and we have Y sperm. When a man, and for the egg, they are all X egg from the woman. But for man or for men, we have X sperm and Y sperm. So whenever X sperm fertilize the X egg, so we give birth to a baby girl. While when the Y sperm fertilize the X sperm, sorry, when the Y sperm fertilize the egg, which is X, it will give birth to a baby boy. So that is what I'm saying. So we have two types of sperm. We have Y sperm and we have X sperm. So if Y sperm fertilize the egg, will give birth to a baby boy. And then at the same time, if the X sperm fertilize the egg, will give birth to a baby girl. So that is what I want you to understand. So, but currently, due to the problem of evolution, mutation is taking place. So the X sperm now, they are changing babies, through the babies that we are giving, through the babies and the our children that we are giving birth. That evolution is taking place, trying to change the Y sperm to X sperm. So if the Y sperm is completely changed to X sperm, it will reach to a stage where there will be no longer, we cannot give birth to a baby boy. And this is what actually, this is exactly what is taking place. This is experimentally proven. It has been experimentally proven that Y chromosomes is undergoing evolution, trying to change to X sperm. So at the end, the men will end up of having X and X sperm. So there will be no longer Y sperm. So therefore, we'll be giving birth to only baby girls, and it will reach to a stage where there will be no longer men on earth. Please, do you understand, ladies and gentlemen? So this is a question that I asked you today early in the morning. That why is it that the number of the girls now are more than the number of the boys? This is because currently the Y sperm is undergoing evolution. So that is what happens. So this is experimentally proven. This is experimentally proven. Yeah, of course, Madhu Divine is started from genetic mutations. It started from genetic mutations, and genetic mutations lead to evolution. And it is that genetics evolution that lead to the change of the Y chromosome. Okay, Letitia, thank you very much for joining the YouTube. So you are welcome. So this is what we call, okay, this is the reason why we give birth to more of baby girls than the baby boy. So that is what actually happens. So this is experimentally proven. It is known already. It has been experimentally proven. I think let me just show you some demonstration. So that is why if you can now check on internet, you can now check on internet for the issue of this change in the chromosome. That is the evolution of Y chromosome. You can check it from the internet. You see, this is X chromosome. I think you can see it. So if you can now check, you will now start seeing something like this. You'll start seeing the Y chromosome moving like this. So it will keep moving. As the time goes on, it will keep moving. I don't know if you are seeing it. You see, this is Y chromosome, as time goes on, it will keep changing, moving, and it will reach to a stage where it will change completely to X chromosome. So at the end, we'll end up having something like this. So we'll have the complete X chromosome. So therefore, it will reach to a stage where there will be no Y sperm. So therefore, there will be no longer baby boys. So I have, yes, it is already proven scientifically. So ladies and gentlemen, this is what happened on that. So now the next thing I said I send you, this is evolution. Things are changing. The human species, not only human, generally a lot of organisms are now changing. It's not only human. It's not only human. So many organisms are changing, but it started from, remember, let me actually take you a little. So it might be permanent. Yeah, of course, it will be permanent. I think for those of us that are Muslim, I think if you can remember, one of the sign of the judgment day, it will reach to a stage where the number of the men on earth will be far more than the number of women on earth. So this is actually what happened. So it started from here. So the explanation is evolution. So we know that before evolution takes place, there must be driving force. So I think I answered this question. So now the next thing that we are going to talk about, ladies and gentlemen, is we are now going to revise. I send you a video link, 3 videos link. So we are now going to move and discuss each and every one of them. So I started asking you to define biochemistry, and I can see, okay, sir, do you mean that they will forthcoming hermaphrodites in the future? We're not talking about, it's not hypothesis, it's not hypothesis. Hypothesis is something that you are just guessing. So it is an experiment that has been proven. It has, I think you can check from the internet. If you want, I can even get the paper and send it to you. Yeah, of course, it will reach a stage where we'll have only female, but I don't think we'll reach up to that point. So hypothesis is verifiable assumption. So I think that is what we discuss on this, and I think it's a question that I asked you in the morning, and I said that I will address it when we come for the discussion. So now, ladies and gentlemen, I now ask you to define biochemistry. And as I can see, some of you have started that biochemistry. Okay, let me ask you again, what is biochemistry? What is biochemistry? Good evening, all. What is biochemistry? What is biochemistry? Okay, biochemistry is a branch of science that deals with the application of chemistry to the study of biological process. Okay, biochemistry is the application of chemistry in the study of biological processes in cellular and molecular level. So yeah, I think you are correct. When we said biochemistry is one of the branches of science that use the application of chemistry to study biological processes at cellular and molecular level. So in biochemistry, we are trying to study the chemical processes that are taking place at cellular and molecular level. So that is whatever we are doing in biochemistry, we are trying to study life at molecular level. And those molecular level that we are talking about, they are carbohydrate, as we discussed yesterday, carbohydrate, proteins, lipids, and nucleic acids. So that is biochemistry. So now let me ask you, the branch of biochemistry that deals with the study of informational macromolecules is called what? The branch of biochemistry that deals with the study of informational macromolecules is called what? It's not immunology. The branch, excellent, molecular biology. Molecular biology is a branch of biochemistry that deals with the study of informational macromolecules, and examples of informational macromolecules include RNA, protein, and nucleic acid. So then, a branch of biochemistry that deals with the study of how a cell or a part of cell is used to produce a product of industrial application is called what? The branch of biochemistry that deals with the study of how a cell or a part of a cell is used to generate a product or to produce a product of industrial application is called what? Excellent. The answer is biotechnology. The answer is biotechnology. So what do you do in biotechnology, ladies and gentlemen? Remember, now, if you have a pregnant woman, a pregnant woman, and she can't deliver a baby, what are the other possible ways as a nurse or as a medical doctor do you think you can explore without CS to bring the baby out from the mother? What do you do? Assuming now you have a pregnant woman under labor and she tried to give birth to the baby, but she couldn't do that. So what are the possible ways do you think you can apply for the woman to give birth to that baby without doing CS? Okay, someone said episiotomy. What is episiotomy? What is episiotomy?
Okay. So I think you can hear me now. So what I'm saying, maybe if you have a pregnant mother and she want to deliver a baby, I thought that maybe you can use insulin injection or adrenaline injection. So that is what I'm saying. Are you not using insulin? Are you using insulin or adrenaline injection? Which one are you using? Excellent. So I think you use oxytocin because oxytocin is used for uh, contraction and relaxation. Sorry, I want to say oxytocin, not insulin, oxytocin injection, and also adrenaline because adrenaline is used to produce energy while oxytocin is used for opening of, or I think when we said oxytocin is used for contraction and relaxation of the uterus. So now the question is, please, have you ever sit down and ask yourself that how the oxytocin and how this adrenaline are generated? How are they produced? Have you ever sit down and ask yourself this question? Because of course you know that oxytocin, it is a love hormones that are usually found in men or in women. They are generally found in human. So now the question is, it that now if I want to get oxytocin, I will get someone, cut the adrenal gland and get the oxytocin? Is it how to do it? Excellent, 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 excellent. Someone just answered the question. They are produced through biotechnology. If you can remove my hair, if you can remove my saliva, you can get the DNA there. And from the DNA, you can get the gene. And after, when you have the gene, through this biotechnological method, you can get the oxytocin. So using bacteria, you can get the oxytocin, you can get the adrenaline. So that is the application of biotechnology. You use a living organisms or some part of a living organism to generate or to produce a product of important, or to produce a product that is important or that are benefits to humans. So that is biotechnology. So you use a cell or a part of a cell. When we said a cell, it means the entire cell, like bacteria, or you can use a part of a cell, maybe a nucleus, to produce a product. Yes, hair and saliva, you can get it. You can get it from sperm cell, you can get it from our sweat and everything. You can get the DNA from there. So when you get the DNA, you can get the gene, because in the DNA is a language of life. So like in human beings, there are like about 25 genes, and each 25,000 genes, and each of these genes is responsible for producing a particular protein. So oxytocin is a protein and also hormones at the same time. So therefore, if you can get the skin, if you can get the hair, you can isolate the DNA, and from that DNA, you can get the genes that you are targeting. So after you get the genes that you are targeting, by just putting it into a bacteria, the bacteria will convert the gene into oxytocin. And of course, the bacteria, they have high generation time. Within 1 minute or within 2 minutes, they can generate to produce millions of them. So if they are carrying the same insulin, they will be they actually multiply themselves, and from there you can harvest a large quantity of the oxytocin, and then you purify it, you package it, and you start selling it as a drug. So that is biotechnology. So it's not that okay now insulin injection, that is for those that are having diabetes, you will just go and cut the pancreas of maybe that individual or someone that is, let's say, uh, maybe okay, someone that is not feeling fine, or you just start trafficking human organs like oxytocin. You will get someone, cut the pancreas, then isolate and purify the insulin. So that is not how to do it. So if you want to produce insulin industrially, the best thing to do is to use a hair. In fact, sometimes some of this you can even get them from plant, but the most easiest way to get them in large quantity, to get them in the quantity that you are interested, is to apply the knowledge of biotechnology. So that is why biotechnology is one of the interesting area of biochemistry, where you can exploit the knowledge of the biotechnology to produce a product of industrial scale. So I hope that is clear. Then the branch of biochemistry that deals, yes, so insulin is gotten from animal gland as well. Yes, I agree. But to get the animal gland is tedious. So do you need to be always slaughtering animals, to be always slaughtering animals to get the insulin? No. There are even some microorganisms that produce insulin, even some plants. But the best way to produce them in large quantity is to apply the knowledge of biotechnology, or to apply the tools in biotechnology. So then another thing, the branch of biochemistry that deals with the study of drugs, the drugs consumption, the drug distribution, the metabolism. In fact, when I say drugs, it means the entire pharmacokinetics, the pharmacokinetics and pharmacogenomics. You know that we take drugs, and when we take drugs, we usually we take drugs, and after we take the drugs, we metabolize them. So which of the branch of biochemistry is associated with that? It's not medical biochemistry. It's not medical biochemistry. The study of drugs and the adverse effects of the drugs and the chemical substances is not pharmacological biochemistry. The answer is toxicology. That is toxicology, not pharmacology. Because drugs is toxic. We don't need it in our body. So toxicology is a branch of biochemistry that deals or associates with adverse effect of chemical substances, and the chemical substances can be drugs. So now let me ask this question, ladies and gentlemen. Where do we metabolize drugs? Where do we metabolize drugs? Which organ? In which organ do we metabolize drugs? Excellent, liver. So specifically, what is the name of the liver cells? The name of the liver cells is called what? The name of the liver cells is called what? The name of the liver cells is called what? Excellent, hepatocytes. Hepatocyte is the name of the liver cells. So specifically, who can tell me in which organ, in which organ do we, sorry, in which organelles, in which organelles of the hepatocytes, in which organelles of the liver cells do we metabolize drugs? The organelles. Endoplasmic reticulum, we have two types of endoplasmic reticulum in a cell. We have smooth endoplasmic reticulum and we have rough endoplasmic reticulum. So which one? Which one? Excellent, smooth endoplasmic reticulum. So that is what we do in toxicology. First, the drugs that we are taking generally they are toxic, and when we take drugs, the best thing is to eliminate them from our body as soon as they elicit their pharmacological activities. So we don't need them. Once they elicit their pharmacological activities, so they have to be excreted. So that is toxicology. So we need to know the organs where they are metabolized and where they are going to elicit their pharmacological activities. So this one is very important. Okay, now let me ask you this question. Do you know the reason why sometime? Do you know the reason why sometime to take a drugs for a specific disease, sometimes you will see that you will be given like two different drugs. So do you know the idea and the reason why sometimes if you want to cure for a particular disease and then at the end you will be given maybe three different medications? Do you know the reason why? Why is that? Why? Sometimes if you are having any problem with some disease, when you go to the chemist, you will be prescribed with three drugs, four different drugs. Why? Someone said for activation. No, I didn't accept this. Argumentation. When you said argumentation, what do you mean with argumentation? Akenwa. What do you mean with argumentation? Someone said for synergistic effects. Yeah, I can agree with that. Potential effect. Yeah, I think I can also accept that. Some is to reduce the adverse effect of the odor. Brilliant, brilliant, Lillian. Lillian, this is the correct answer and it's the best. The reason why sometimes we take three drugs or more than that, if you take eight drugs, maybe drugs A will cure the disease, but because it has a side effect, maybe it can cause another problem, then the drugs B will take care of it. And the drugs B will take care the effect of drugs A and it will cause another effect while the drug C will take care of the effect of drug C. So this is what happened. Excellent to reduce the side effect. But sometimes some drugs are synergistic. They have the same role. Yeah, it's not that it might not be harmful, it might be harmful, but maybe the harmful effect of the C might not be that very deadly. But truly speaking, taking drugs is dangerous. Taking injection is dangerous because they are toxic and we don't need drugs in our body. It's because there is no any alternative and there is no any option. That is why we are taking them. And very importantly, ladies and gentlemen, yeah, Aisha Rabiu Aliyu, I can also accept this to prevent drug resistance because sometimes there are some drugs, if you are taking them, they cannot clearly clear the parasites or cannot clearly remove the, yeah, they cannot clear the whole parasites. So that is why the best thing, you need more, so that at least once you take the drugs, they will clear the parasite directly. But if you are taking drugs that cannot completely clear the parasite, the problem is that the parasite will begin to sense the drugs because they have high generation time. So before you know it, within a shortest possible time, they will develop resistance against that drugs. So that is why the best thing, if as a patient, if you are taking a drugs and if a doctor prescribe a particular drugs for you, please take it and keep taking the drugs until when you are really sure that the parasite is completely eliminated. Because if you start taking drugs, after 1, 2, 3 days, the parasites begin to clear, but you stop taking it. So some of those parasites that are not completely cleared, they already sense the drugs. So as they are generating, or as they are producing more of their type, they will be sensing, sorry, they will be sending the same message, the same sense that they sensed for that drugs to their offsprings, and that will lead to the mutation. Remember, Alexandra Fleming is the first scientist that developed antibiotics in the year 1941. But before you know it, in the year 1943, the parasite become resistant to that drug. So see all these things that we are doing, I'm just because there is a video that I send you. So the interactions, most of the interaction that we are going to do, we are going to be more of application. We are going to be more of application. That is to give you the applications of what you watch from the videos. So please, I hope, are you following? Are you following, ladies and gentlemen? Are you following me? Okay. So now the next thing from the videos that you watch, please, I send you so many videos. So I want you now to ask me questions based on what you have watched from those videos. Please ask me questions. Ask questions. Based on the videos that you watched, I want you to ask me questions. I want you to be asking me questions based on what you watch. Okay, sir, please ask us questions instead. Okay, do you prefer? Okay, you are asking, sir, isomerism of carbohydrates. No, we didn't come to that, please. We didn't come to isomerism of carbohydrates. Excellent, excellent. So someone asked question because I know that part of what I discussed. Sir, please further explanation about Human Genome Project, please. Okay, Human Genome Project. It is a project that's about 5 different countries sequence the whole human genome. But I think I need to explain what is human genome. You see, when we said human genome, it means our own DNA. So our DNA is large. And let me tell you, ladies and gentlemen, the Human Genome Project is what solved a lot of problems. It is the Human Genome Project that lead to that lead to a lot of discovery, like the CRISPR technology. It is through the knowledge of Human Genome Project that now we have designer babies. When we say designer babies, they are scientists that, as a father, you can go and meet them and ask them, this is the kind of baby that you want. You want a baby with good nose. You want a fair girl. You want a tall baby girl. So we have designer babies. So it's through the knowing, or it's through the Human Genome Project, that a lot of diseases are diagnosed. Before Human Genome Project, nobody have a knowledge of what sickle cell anemia is all about. Nobody, nobody will tell you or give you knowledge of what sickle cell anemia is all about, especially the disease associated with genetic diseases. Nobody. Someone will just tell you, I there are genes, aljanu. There are genes, I aljanu ne. Because at that time, there is no full knowledge of what human genome is all about. So, what is human genome? As we said, we have a DNA, and we have DNA in the nucleus, we have DNA in the chloroplast, and we have DNA in the mitochondria. So, a human genome project is a project that trying to translate, or they're trying to get the information of what are the sequence of the nucleotide, because what form the DNA is the nucleotides, and those nucleotides are adenine, guanine, cytosine, and uracil. So these are the four nucleotides. And do you know that, ladies and gentlemen, if you take the DNA, the human DNA, if you stretch it, if you stretch the human DNA, if you stretch the human DNA, if you take the human DNA and you stretch it, the length will be between the surface of the earth to the sky. If you take the DNA of one cell and you stretch it, it's more than 6 feet. It's more than 6 feet. So that is why. But you will begin to ask this question that the DNA of one cell is up to 1 6 feet. But now the question is, how is that DNA, that small DNA, how does small DNA get itself into the nucleus? Into the nucleus, the cell that you cannot even see it with the naked eyes, even the cell itself, talk less of the nucleus itself. So what usually happen? The DNA, it usually twisted itself, wound itself like a normal thread. So that is why, because it is twisted, it is rubbed, wound itself together, because it is very thin but very long. So it's like a normal thread. So it wound itself, round itself around a histone protein, and that is why it perfectly fits into the nucleus. That is why it is perfectly fit into the nucleus. So it is that human DNA that was sequenced. Those letters, those four letters, adenine, guanine, cytosine, and thymine. So those four letters are the four letters that constitute or that rearrange themselves through permutation and combination, and that is how they are. So as a result of that, because it is very long and one lab cannot know when you said human genome sequence or human genome project, it means to know how the nucleotide sequence are arranged, the sequence of the nucleotide that are present in that DNA. So that is why, because the DNA is huge, so it coated it, the DNA was fragmented into different fragments. Some part of it was taken to Germany, another one UK, US, China, and Japan. So they are taken to 5 different labs across the world: China, Japan, US, UK, and US. So they are taken to this lab. And this human genome project it started since 1990, and they begin to sequence it. They begin to sequence it. That is to know the sequence of the nucleotide. It took them about 13 years. So the Human Genome Project is started in 1990 and ended in 2003. So yes, France, excellent, France and France. So these are the 6 labs that carried out this Human Genome Project. And do you know that after the sequence the human genome, it found out that in human genome there are about 3 billion base pairs. 3 billion base pairs, that is the number of nucleotides that are found in human DNA. Please, do you understand? So after this human genome project, we know the number of the nucleotides that are found in our DNA. And we know the number of genes that are found in our DNA. 3 billion base pairs, meaning 3 billion nucleotide sequence. So this is how the Human Genome Project was conducted. And very importantly, ladies and gentlemen, that is when we know that we have 3 billion base pairs, and also we have about 25,000, more than 25,000 genes. And each of that genes is responsible for producing a particular protein. Like, for example, is after the Human Genome Project that we know that there is a hemoglobin gene, and it is responsible for producing hemoglobin as a protein. And that hemoglobin protein is of different types. We have hemoglobin S, we have hemoglobin AS, and we have hemoglobin SS. And what is the difference between this hemoglobin? The differences between this hemoglobin is that is because there is a change. There is a change in some nucleotide at some position, and that is what we call mutation. And that's what leads to the sickle cell disease. So as a result of Human Genome Project, a lot of diseases were diagnosed, a lot of diseases were cured. The issue of vaccine development, the drug development, the disease diagnosis, and everything came as a result of this Human Genome Project. Currently, now there is a CRISPR technology. A CRISPR technology is a technology that, okay, you can check, okay, a mother conceived and you want to see if that mother is carrying a baby with some genetic defect. So you can get an aliquot of the DNA, maybe from uh, what you call amniotic fluid. You can get the aliquot of the DNA of the baby. You can check through the DNA and see if there is any genetic problem. You can use the CRISPR technology to correct the mistake. And it is this CRISPR technology that if you have someone that is having sickle, with CRISPR technology, you can edit the gene. Please, ladies and gentlemen, I hope it is now clear. Please, do you understand the Human Genome Project now? CRISPR technology, CRISPR technology. I will write it and yes, it's edit the gene. It's edit a gene. If there is any genetic defect, you can use a CRISPR technology, edit it. It's not a crystal, CRISPR technology. CRISPR technology. CRISPR technology. Check it. I wrote it on the comment section now. Look at it. Look at my comment. CRISPR technology. Okay, please, ladies and gentlemen, as I mentioned, if you know that you are if you know that you are not a member to my YouTube channel, I'm not teaching only biochemistry, but I'm teaching my student biochemistry. But in my YouTube, there are also other courses. There are other courses like physiology 225, physiology 227, biostatistics, and we also have anatomy videos. So I have all my videos on these topics, and if you want to watch them and you want to have access to them, you have to register with the YouTube membership. And as I said, the biochemistry videos, they are all free. They are all free. You don't need to make any payment if you want to access all biochemistry videos. They are all free. But for my uh, human physiology 225, human physiology 227, biostatistics, and anatomy, you need to make a payment. And the payment is just 1,500, is not much. And you will have access to all the videos. Once you make that payment, you will have access to all these 5 courses. But for biochemistry, is free because you are my student and I am assigned to teach you. So all the biochemistry videos, they are free. So if you even come across any videos that require payment for biochemistry, so please alert me, it's a mistake. So I will convert it to free video and send it again. Please get this, very important. And also for the other courses, we do a discussion like this. And sometimes if you are not a member, you cannot even have access to the lecture. And we solve first question papers for the biostatistic, for the human physiology 225, and the anatomy courses. So we solve first question paper. So that is what I want you to understand. So that is why now I'm saying if you want to register, it's just to use your ATM card and make payment. If you can check, you see Adolari Adejoke. So it means that she is already a member. That is why you see her name different from your own. Yeah, you just use your ATM card. You see join. So when you see that join, you click on the join. So once you click on the join, it will take you. You will see 750. You will see 1,500, and you will see 4,000. So I'll advise you to either register with 1,500 or 4,000, so that at least you can have access to all the videos and also the past question papers that we solved, and we are still going to solve more past question paper. So you can just use your ATM and register with the YouTube. I okay, someone is the child will be okay now that it is what is being used for IVF selection. Yeah, the child will be okay after you edit the gene. So I tried making payment through my card, but it wasn't going. You can, if you try your ATM card and it didn't work, you can use your friend ATM card and pay the money. It will work. It will work. Use your friend ATM card. Okay, I think I will. If you, I hope you are. If you are not in my tutorial group, there is a tutorial group where we have a timetable and we are doing all these courses. Transfer is not going. YouTube or Google do not accept payment with transfer. So we have a timetable in the other group. We have a group tutorial where we are doing all these 5 courses. So if you are a member and you are also interested in the tutorial, you can join. Yeah, you can become a member if you have ATM, and you can, when you go to the YouTube, after you subscribe, you'll see joined. So click on the join, then it will take you where you will make the payment. And what I want you to understand, please, I want us always to differentiate two things. There is a group for tutorial where if you want to register with this YouTube membership, or if you have any problem with the membership, that is why we are going to talk. But if you have problem with Biochem 241, you are going to discuss that on the group that is specifically meant for it, please. Even now that we are doing this lecture, you can even see. So on the timetable today, we have human physiology 225. Yes, of course, I sent the video for human physiology 225. And tomorrow in the timetable, we don't have anything. So that is why, because now we are doing this biochem 241. So tomorrow as Monday, we are going to discuss those videos on those videos of the human physiology 225. So tomorrow we'll do that. Maybe we'll even have a similar interaction like this. Yes, the 4K is yeah, you can access all the videos if you pay 4K. So you see Amina Abdul just joined. You see, so that is how to do it. Amina Abdul just joined. I think there is also another person that joined when we started the class. So I hope now you understand the concept of Human Genome Project. So what other questions do you have, please? Apart from the Human Genome Project, what other questions do you have? So after subscribing, I didn't see any join. No, you have to see it actually. You will see it. Okay, I think after this I will send a link on how to join to the group. So once you click on the link, it will take you directly where to join. Okay, so someone is asking what is the difference between DNA and RNA. So actually at this level, we are not going to talk about that. The difference between DNA and RNA, there is a video, and also we are going to have a lecture specifically on the DNA and RNA as well as their differences. Yeah. But one thing that DNA is double-stranded and RNA is single-stranded. And the RNA are usually produced from the sequence of DNA. And then there are so many differences. So we'll come to that. So please, if you have any questions, okay, throw more light on structural biochemistry. Okay, so thank you very much. Someone is asking about structural biochemistry. So structural biochemistry is one of the branches of biochemistry that is usually associated. So you see another one, Priscilla Chikwe, she just joined. So a structural biochemistry is a branch of biochemistry that is associated with mostly discovery of drugs or drug discovery. Like for example, most of the drugs that you are seeing, they are enzymes inhibitors. So part of this lecture or part of this course, I will teach you on how to come up with a new drugs. Because generally drugs are enzymes inhibitors. So some of the enzymes, their structure is not known. So in structural biology, you can isolate the enzyme. After you isolate the enzymes, you can crystallize the protein. After you crystallize the protein, you can make it into a crystal form that can be easily considered and for drug development. So structural biology is associated with discovering the structure of new proteins, the discovery of the structure of enzymes which can be exploited for drug development process. And in structural biology, we exploit a lot of techniques and some of the techniques include number one, we have crystallography, we have X-ray crystallography, we have NMR, we have AAS, we have so many of them. So please, what other questions do you have? So all the videos, they are clear. I think they are very clear when you watch them. They are straightforward. So, but we are going to be having discussion. So I will be sending you the videos before any lecture. I will send you the videos, and before the lecture time, you go through the videos, and after you go through the videos, then we will now have a time to come and discuss like this. So each and everything that you need more explanation, then we'll discuss on it and we'll see how you can apply it practically. Yes, Thomas, just street, add it, add your street, and it's not compulsory, it's optional. It's optional. So please, is there any question? Is there any other things that you didn't understand among all the three videos that I sent to you? Any questions? Then the explanation is different what I watch in the videos. Yes, the video is straightforward, but this thing that we are doing is we are doing like discussion. So I am opening it very well, because one thing that I want you to understand, my video my videos on YouTube is a global videos. So many people from other part of the world are watching. I have subscribers and viewers from US, from UK, from UK, from Philippines, almost from every part of the world. But this life is like exploration as in more explanation. Yeah, you have to start first from those videos. So after you watch them, then we come here, and then the reason why I'm doing this is because I wanted you to really appreciate biochemistry and also to use the knowledge that you acquire here and apply it in your daily practices. So that is the essence. So please, for those of you that didn't watch the video very well, please go and watch those videos. Go and watch those videos. And also, in addition, if you are not a member, please, okay, video is saying the structural biochemistry is the understanding of chemical architecture of biological macromolecules, especially proteins and nucleic acid. Yes, that is what we said. That is the chemical architecture. The chemical architecture, that is to know the structure of the biomolecules or the macromolecules. And the macromolecules is usually proteins and nucleic acids. So if you are able to know the chemical architecture of biological macromolecules like proteins and nucleic acid, then you can use that chemical architecture for drug diagnosis, for disease diagnosis and drug development. Someone is asking what are the major branches of biochemistry? Aisha Ghazali. So we have nutrition, we have enzymology, we have biotechnology, we have medical biochemistry, we have immunology, we have structural biology, we have molecular biology. Yes, sir, it is really interesting because at first I was thinking the course will be hard, but I am having different view now. Thanks to you, sir. Truly speaking, biochemistry is not easy. It's not easy. That's just the truth about it. But it depends on how you take it. It depends on if you are reading it and you are watching it or you are committed to it. You will actually find it very easy and very simple. It is much more difficult than any course that you are doing this semester. It is that is just the truth about it. It is much more difficult than any course that you are doing. That's just the truth about it. Yeah. But it's just that I'm doing my best to teach. The videos are available and there is also there are explanation here. So that is it. And I will do my best, and as time goes on, I will also help you with solving past question papers and so many other tricks on how to pass their examination. Yes, immunology is a branch of biochemistry, yeah, of course. So under the macromolecules, it was listed to be protein, lipid, carbohydrate, and DNA, RNA, but you had mentioned that carbohydrate is not part of it yesterday. Carbohydrates, I mentioned yesterday that it is not an informational macromolecules. It's not an informational molecule. Informational, but the carbohydrate is a macromolecule. DNA and RNA as well as proteins and lipid are all macromolecules, but protein and nucleic acids, which include RNA and DNA, are informational macromolecules. So thank you so much for your explanation. I think it's sinking small, small. So thank you, Gloria. So I think if there is no any questions, I think we are going to stop here. We are going to stop here. But I'm advising you, if you are not a member, try to be a member because don't delay it. Don't delay it. I'm just advising, don't delay it. Because as you delay it, you will be missing something. You will be missing. Like tomorrow, we are going to do physiology. Next tomorrow, I think is biostatistic calculation. And so please try as much as possible to join so that at least you will not be delayed. So thank you very much, ladies and gentlemen. We are going to stop here, and please don't forget to subscribe the YouTube so that at least once you subscribe, you can have access to the videos all the time. Thank you very much and see you next. Bye bye.